Okay, good morning. I'm very happy to be here. My name is Sean Leng. I'm also a geriatrician. I see many familiar faces here. So I give this talk for Joe Markwick. Joe is not be able to make it here. Actually, Dr. Peter Hunt's talk just set a stage for my very brief talk. So can I get to the next slide here? So in terms of background, we know the people who live with HIV has a very high prevalence of CMV infection. We actually measured the anti-CMV IgG antibody concentration for the entire max cohort. So we have we do have a poster outside, it's paper number 50, to really present all this CMV IgG titer data at the baseline of the cohort. Here we'd like to talk about the impact of the antiretroviral therapy on the concentration of the CMV IgG titer. So we know, really, the CMV infection can be linked to chronic inflammation, as Peter mentioned, and also not only in the people living with HIV, but also in the general geriatric population. Now, really the question here, whether they, in the people with HIV infection, whether the antiretroviral therapy would have any impact on the CMV IgG titer is not fully understood. So that's why we have done this. We have done this study. So in terms of methods, we really look at 163 people in the max cohort who were HIV positive and undergone anti-CMV and antiretroviral therapy. What we wanted to do is we want to make sure with the antiretroviral therapy has the full HIV suppression. So we will look at the anti-CMV IgG titer, both at the baseline, but also at the visit before the antiretroviral therapy started. But then we look at the third time point when we know the HIV viral load has been suppressed for 2 consecutive visits. So that means 1 whole year. And then we also, look at the last visit, when the participant actually still, in the study up to 2019. So the anti-CMV IgG titer measurement, we use the assay as I listed in there. But then we also build in the the WHO international standard, so that we can look at the international unit per ml. So that's the concentration. And actually, the other purpose we want to use that international standard is if other studies use the same standard, then we can compare the results between different studies. So here is the data, actually the baseline of the study population. We listed the 2, both in terms of participants in our study, but also those who were not, who were excluded from this study. So to see whether the actually general, the general representation of the entire cohort. So as you can see, actually the ones who were in the study have a longer follow-up period, and then also obviously with the for HIV suppression has less AIDS diagnosis, and other than that, they're pretty similar. And then here is the main data data table. I know it's a very busy one, but I want you to focus on the left side of the the entire study population. So what it shows there is look at the actually 3 follow-up period. So that means before the antiretroviral therapy started, and then also between, but then after. And so you can see, the follow-up period for each of those, each of those study, study intervals, and then, but also the change of the anti-CMV IgG antibody concentration. But then also we try to annualize it. So you can see for the first 2 study intervals, the annualized rate is about about 2 international units per year. But then, except the last one, which after the full suppression of HIV, achieved, which is really flat. But then the right side of the table is really looking at the, so out of those 263 individuals, we have 198 who were diagnosed at the baseline. That's the HIV prevalent. But then has about 60 some actually came along, became HIV positive. So that's the HIV incidence. But then the 2 groups were, no difference. So here just to highlight, the same table, just highlighted in red. So in terms of the annualized change, basically what shows here actually shows on the next slide is the figure. So here I want to orient you guys is, the x-axis is the log transformed anti-CMV IgG titers, right? So actually that's the y-axis. The x-axis is the 4 different time points I mentioned, and you can clearly see now the dark line is the overall, but then the red line above is the HIV prevalence group, and then purple line is the incident group. So you can clearly see the first 2 intervals, without HIV suppression, the CMV antibody concentration, rises. But then once the HIV suppression is achieved, then it's really flattened out. So this really shows, this longitudinal study shows you the trend. So in conclusion, really suppression of the HIV replication by antiretroviral therapy leads to stabilization of this, circulating anti-CMV IgG concentrations. And, this finding may suggest that the, really improved control of the CMV infection. But then this could be, this is, could actually suggest a, actually the reduction of the contribution of the CMV infection to, for example, chronic inflammation and other age-related diseases. But obviously more studies would actually needed to test this hypothesis. So I would acknowledge the MAX study, MAX study participants, of course, but then also the funding. Well, I will stop here. Thank you.